All right, so today we're going to talk about the reproductive system, and this system is very unique to the body compared to the other 10 systems we've been looking at in the body. It's unique because it's not essential for life. So it's not like the cardiovascular system or the urinary system or the lymphatic system. It's not essential for life, for living. It does affect other systems, though, and we'll take a look at that. So first of all, we're going to look at the male and the female systems separately because they are very unique and different from one another. The male and female reproductive systems produce and store specialized reproductive cells called spermatozoa in the male and oocytes in the female. And these cells combine together to form a new life. In addition to this, the organs of the male and the female reproductive system produce hormones. And these hormones play a major role in maintaining normal sexual function. So the spermatozoa and the oocytes, those are both called gametes. The gametes are produced by organs that are called gonads. The gonad in the male is called the testes, and the gonads in the female are called the ovaries. From these gonads are ducts, and the ducts carry these gametes into the reproductive tract. The reproductive tracts eventually then lead to the exterior of the body. And along the way, accessory structures are going to secrete fluids into that reproductive tract. Before we get started looking at the male reproductive system, let's look at some differences between the male and the female. So the male produces a large quantities of gametes or spermatozoa. Actually, they produce about 500 million gametes per day. The females, on the other side, they are going to produce 1 gamete per month. So 1 oocyte per month. The males, the testes secrete the male sex hormones called androgens. The main androgen is called testosterone. And they produce the male gametes, which are called spermatozoa. The gonads in the female are called ovaries. And the ovaries secrete female sex hormones called estrogens and progestins. And again, they produce 1 immature oocyte per month. If the sperm reaches the oocyte, so if the spermatozoa reaches the oocyte, fertilization takes place, and then the oocyte can mature into an ovum at that point. Fertilization continues between the spermatozoa and the ovum, and together they will produce a zygote, and the zygote will implant into the uterine lining in the female, and we'll talk more about that in chapter 29 when we get to development and genetics. So let's take a look at some of the anatomy of the male, and we'll go through the male system, and then we'll get to the female system later. So first of all, we can see that the male has several structures that we're going to need to know. The first one, if we look at the skin down here, this skin is called the scrotum. And so there's different layers of the scrotum, and we're going to talk about that in just a minute. Inside the scrotum are these organs that are called testes, and the testes are covered by a membrane, but here we can see the testes. On top of the testes is another gland that's called the epididymis. So that's this right here. We'll talk more about that. And then leading away from the epididymis is this duct that's called the Ductus Deferens. So it is this line right here that I'm drawing in. Okay, that is, or that I'm highlighting, that is the Ductus Deferens. So along the way with that Ductus Deferens, there are also these blood vessels. So we can see the arteries and veins that go along with it. And there are some muscles, some smooth muscles that wrap around that entire ductus deferens and blood vessels, and we call that the spermatic cord. The spermatic cord will follow the ductus deferens and the arteries and veins up through this opening here in the inguinal ligament. The spermatic cord continues through that opening up into the abdominal cavity, and then it meshes in with the muscles of the abdominal muscles. If we look down at the end of that spermatic cord, we can see that the muscle surrounds the testis. It surrounds that gonad. And then that muscle, when it's surrounding the testis, we call the cremaster muscle. So then we take a look again at the scrotum. So the scrotum has 3 layers. The innermost layer surrounds that cremaster muscle, so that's just some fascia. And then there's skin on the outside of the scrotum that we call the scrotal skin. Now between the fascia and the scrotal skin is another muscle that we call the dartos muscle. So that dartos muscle is right in here. Okay, it's that whitish area in there. And that dartos muscle gives the scrotum its wrinkled appearance. Together, that dartos muscle and that cremaster muscle work together to keep the temperature of the testes slightly cooler than body temperature. So how it does that is whenever the temperature outside is a little cooler and the testes are cooler, they do hang away from the body, then those muscles will contract and they will pull the testes up closer to the body, and then the temperature in the testes will increase. And then in a hot day, when it's too hot, then the testes will need to be further away from the body. And so the cremaster muscle and the dartos muscle relax, and the testes then moves further away from the body. Now it's important to keep the temperature just at the right temperature in the testes because the spermatozoa have to be produced and have to mature in that area, and they can only do that if they're at a temperature slightly cooler than body temperature. Underneath the scrotum is a serous membrane that lines the scrotal cavity to reduce friction. This is called the Tunica Vaginalis. So we can see that right here. And because it's a serous membrane, it's going to have both a visceral and a parietal layer. So this diagram you will most likely have on your lab exam. You're going to want to be able to identify several structures on here. Also on this same diagram, we can see the penis. So we have the body of the penis, which would be right here. Then we have the foreskin, otherwise known as the prepuce of the penis. Right here, it's that flap of skin that hangs down from this expanded area of the penis, which is called the glans of the penis. This will most likely be a diagram on your lab exam. So let's take a look at the testes. Each male has 2 testes. A single organ is called a testis. The testis is egg-shaped. It's about 5 centimeters long. It's about 3 centimeters wide, and it weighs about 10 to 15 grams. Each of the testes lies in their own separate scrotal chamber, and there's a partition in between the 2 scrotal chambers called the raphe and it's a thickening of the scrotal surface. The Tunica Albuginea are fibers in the testes that separate the testes into separate lobules. So here we can see these red lines here. These are the Tunica Albuginea, and they are separating the testis into different lobules. The lobules then contain 800 slender and tightly coiled seminiferous tubules. Sperm are produced in the seminiferous tubules, and in cells just outside those seminiferous tubules called interstitial cells, that's where androgens are produced. And testosterone is the main androgen. We'll see those cells in another picture. During development of the male reproductive system, the testes begin in the abdominal cavity. In the male fetus. And then those testes begin to descend through the abdominal cavity to the scrotum as the fetus starts to develop. So we call this, as the testes descend from the abdominal cavity down to the scrotal sac, we call that the descent of the testes. So the testes are forming inside the body cavity just next to the kidneys. And then during the 7th month of gestation, the fetus begins to develop very rapidly. So the fetus has a lot of hormones circulating around in his body, and it causes his testes to move through the abdominal musculature. As the testes are descending, other structures, such as the Ductus Deferens, which is the tube that sperm flow through from the testes, as well as blood vessels and nerves and lymphatic vessels, they descend with the testes as well. And as the testes are descending downward then, those other structures, which were the Ductus Deferens, and then we also had some arteries and some veins and some lymphatics that are descending along with them. All of those structures then start to form the spermatic cord. And then the testes moves through the inguinal ligament and into the scrotum. The spermatic cord is surrounded by both muscle and fascia. And the scrotum then is the fleshy pouch that is suspended just below the surface region between the base of the penis and the anus. And the testes then sit right in each of those scrotal sacs. So we can see the testes again on this diagram. So here is the scrotum, and the scrotum is made up of skin, and then the dartos muscle, and then the fascia. Inside the scrotum, we have the cremaster muscle. Then there is the tunica vaginalis, which is that serous membrane. So we have the parietal layer, and then we have the visceral layer, and then in between we have that cavity, the scrotal cavity, where the serous fluid is, so that reduces friction. Inside the tunica vaginalis, we have the tunica albuginea, which is right here. And then there are partitions from the tunica albuginea that extend up into the testes and create lobules. And in the lobules are the seminiferous tubules. So those are those purple-looking tubes inside each of the lobules of the testes. The seminiferous tubules are going to all converge together in another structure called the rete testis. The rete testis is a small passageway of tubules that are produced from the convergence of all those seminiferous tubules. So here's the rete testis, and then the rete testis has a couple of passageways that leads into the next structure, which is called the epididymis. And the epididymis then leads into a single passageway that is called the Ductus Deferens. So these sperm are produced in these seminiferous tubules, and then they flow into the rete testis. And from the rete testis, they flow into the epididymis, and from the epididymis, they flow into the Ductus Deferens. So let's talk about spermatogenesis, which is the production of sperm. Spermatogenesis occurs in the testes, in the seminiferous tubules, and they occur in these cells that we call the sustentacular cells. So that's where we're going to start today. Let's take a look at one of the cells. So this is one of the cells that's lining the seminiferous tubule. So if we take a look, say, at a cross-section of the seminiferous tubule, that's what this would be. Seminiferous tubule. The cells that line this seminiferous tubule are called sustentacular cells. So each one of these would be sustentacular cells right there. A more common name for them are just nurse cells, but we would call them sustentacular. And so what I'm going to do is I'm going to show you one of these cells. So here's one cell, and I'm going to take that cell and draw it right there. So now you can see the sustentacular cell. Yet another name for this sustentacular cell is also called the Sertoli cell. And the sperm grow inside these cells. So let's take a look at one of these nurse cells. So here's the nurse cell, and here's the nucleus of the nurse cell. And when the FSH is secreted from the anterior pituitary, it will stimulate this process of spermatogenesis. So I'm going to show you what it looks like inside the nurse cell, what goes on. So just so we can get our bearings here, if we go back and we look at this seminiferous tubule, inside the seminiferous tubule is the lumen. And so this lumen is filled with a fluid, right? So the lumen on this picture of just this cell would be out here. So in the deepest layer of this nurse cell, or Sertoli cell, we see Spermatogonia. Now, Spermatogonia are stem cells. And what stem cells can do is they can divide, and then their daughter cells can differentiate into a cell with a specific function. So the Spermatogonia have the same number of chromosomes as all the other somatic cells in your body. They have 23 pairs of chromosomes. In a pair, one of the chromosomes had come from the father, and one of the chromosomes in the pair had come from the mother. And so there are 23 of these pairs. So in a pair, the chromosomes are going to code for the same thing. So in the paternal chromosome, this chromosome, there might be a gene in there that codes for, say, hair color. And maybe the gene for that hair color that came from the father says that the hair color should be brown. And the other chromosome from the mother would code for hair color as well. But in this case, since it came from the mother, say it was coding for blonde hair. Now there are 23 pairs of these chromosomes. So you might see other chromosomes that are a little bit shorter, and they're going to code for something else. You might see a chromosome that's even a little bit longer, and they'll be coding for something else as well. So anyway, these Spermatogonia have 23 pairs of chromosomes. just like all the other cells in your body. Now, at the end of this process, we're going to go from a spermatogonia to a spermatid. And by the time we get to the spermatids will only have 23 chromosomes. So no longer do they have the pairs. And so we'll end up having 4 spermatids, and each one will contain 23 chromosomes. So half the number of chromosomes as the original cell. We're going to talk about the processes of mitosis and meiosis and how the one spermatogonium becomes 4 spermatids. And each spermatid has only 23 chromosomes when the original spermatogonium started out with 23 pairs, so twice as many. So before we get into the process of mitosis and meiosis, I want to show you the phases that occur as the spermatogonia becomes a spermatid. So here we have the spermatogonium. So spermatogonium is singular, so we have one cell. It's in the deepest layer of the nurse cell, and it has 23 pairs of chromosomes. So the first thing that happens is this process called mitosis. So we know mitosis. It has 4 phases. It has prophase, metaphase, anaphase, and telophase. And at the end of mitosis, we end up with 2 daughter cells that are identical to the original cell. So this spermatogonium goes through this process of mitosis, and one of the daughter cells gets pushed up 1 layer closer to the lumen in the nurse cell. And now this cell is called a primary spermatocyte. The other daughter cell is going to remain in that deep layer of the nurse cell, and it will continue to be a spermatogonium. So we're going to talk about what happens then to that primary spermatocyte. The primary spermatocyte then will go through the process called meiosis 1. And meiosis 1 is a very special kind of mitosis. So we have the same phases. We have prophase, metaphase, anaphase, and telophase. But before this primary spermatocyte even enters into meiosis 1, it has to go through an interphase. And so we'll call this interphase 1. And this, of course, is where the DNA are going to replicate. So we get DNA replication, and we get twice as many chromosomes. The DNA are making copies of themselves. And then the cell undergoes meiosis 1. So meiosis 1 has, again, like I said, prophase, metaphase, anaphase, telophase, and we're going to actually end up calling it prophase 1, metaphase 1, anaphase 1, and telophase 1. That lets us know that we're actually in meiosis 1. At the end of meiosis 1, we end up with 2 cells. Now these cells are called secondary spermatocytes. So here's one of the cells, and here's one of the other cells. Both of these cells are going to undergo the next process. The next process is called meiosis 2. So meiosis 2 goes through the same prophase, metaphase, anaphase, and telophase, and we'll just say prophase 2, metaphase 2, anaphase 2, and telophase 2, so that we can distinguish it from meiosis and mitosis. Now, one distinct thing is that between this, right before the meiosis 2, when the secondary spermatocyte starts to develop further, there will be no interphase. No interphase right there. So we won't have any DNA replication. So there's no DNA replication there. And at the end of meiosis 2, then, each one of these cells is going to give rise to 2 other cells, right? So 2 other cells, and then this one is going to give rise to these 2 cells, right? So we end up with 4 cells total. So the secondary spermatocytes now become the spermatids. And we end up with 4 spermatids. We have 2 spermatids coming from this cell and 2 spermatids coming from this cell, right? So there's 1, 2, 3, and 4 spermatids. Now these spermatids still don't look like a sperm. A sperm has not a lot of cytoplasm around its nucleus, and it has a flagella or a tail. But spermatogenesis is over. So as soon as we get to the spermatids, spermatogenesis is over. It ends right there. But like I said, the spermatid still doesn't look quite like a sperm. So the spermatid has to develop into these sperms. So each one of these spermatids will develop into a sperm. And they do that through the process that's called spermiogenesis. Spermiogenesis. So during spermiogenesis, the cytoplasm gets lost, so there's not very much cytoplasm, and all we have really is a nucleus left, and then it grows a tail, which we call a flagella. So we're going to take a better look at a mature sperm in just a little bit. So now we have these cells. As the spermatids go through spermiogenesis, they end up as these cells with very little cytoplasm and a flagella, and we call those cells spermatozoa. So at the end of spermiogenesis, we have spermatozoa. Now these spermatozoa are what we say they are incapacitated. They are also immature. So we see these cells here, and they're stuck in the cytoplasm of the nurse cell. And it's just their tails that are outside of the nurse cell. They're kind of floating around there in the lumen, in the fluid in the lumen of the seminiferous tubule. So the next thing that has to happen is that these spermatozoa have to be released. So they're released into the lumen of the seminiferous tubule. They become unlodged, and now they're freely floating around in the lumen of the seminiferous tubule. That process of having them unlodged and released is called spermiation. Spermiation. Okay, so they get released, and now they're in the lumen of the seminiferous tubule. So if we go back over here at this picture, now we can see them floating around in there, right? And so they're still incapacitated, which means the flagella can't move. They're still immature, so they have to move into the organ that's called the epididymis in order to mature. So that sperm then will be moved to the epididymis. And how is it moved? It's moved because these ducts have cilia, and the cilia will help to move those newly formed spermatozoa into the epididymis. And then it's in the epididymis where they will mature. So that we're going to talk about in the second video. So let's take a closer look at spermatogenesis and look at the phases of mitosis and meiosis. So first of all, we're going to draw just one of the pairs of the chromosomes. So here we have one pair. This one came from the dad, and then this one came from the mom. All right, so we have got one pair. There's normally 23 pair in this spermatogonium, because that's what this is. This cell is a spermatogonium. And so we know what happens as a cell starts to divide. It goes through DNA replication, and so then we're going to end up with twice as many of those cells. So we have here now we are going to replicate the DNA, and now we have twice as many chromosomes. And again, I'm just showing you one pair. So then during this mitosis, we have prophase, metaphase, anaphase, and telophase. And in prophase, the nuclear membrane disappears, and we get the formation of that spindle apparatus, right? So we get the spindle apparatus forming. And then in anaphase, we see how these chromosomes are going to separate. So then as they separate, we have one cell that ends up with the pair, and then we have the other cell that ends up with the other pair. So now we have two cells, we have two primary spermatocytes, and they look identical to that original spermatogonium, right? So the one cell here is going to remain as a stem cell, so it remains as a spermatogonium. And the other cell moves up a layer closer to the lumen of the seminiferous tubule. It's identical to the spermatogonium, and we call it a primary spermatocyte. So here we see one of the pairs. So this is one of the 23 pairs of chromosomes. So the next thing that's going to happen is this primary spermatocyte is going to go through interphase, and this is going to be called interphase 1. So the main thing that happens in interphase 1 is that the DNA replicate. Same thing. So we're going to see how we have one chromosome from the father is going to replicate, and the chromosome from the mother is going to replicate. So we end up again with double the amount of chromosomes. Now the cell is ready to go through meiosis 1. So in meiosis 1, what's going to happen here is we're going to have the prophase 1, the metaphase 1, the anaphase 1, and the telophase 1. In prophase 1, the nuclear membrane disappears and the chromosomes bind together. Now in mitosis, what we saw was the father's chromosomes, their copy and the copy, they bind together. And then we see that the maternal chromosomes from the mom, they also bind together and they form these things that we call chromatids. So that's normally what we saw in prophase 1. But another thing happens in meiosis 1 during prophase 1 that's really important. So these two chromatids in meiosis 1, they join together. So the process of them joining together is called a synapse. And what they form is called a tetrad. So this happens in meiosis 1 during prophase. So it happens during prophase 1. And it's really a unique thing that happens because after this synapse occurs where they join together like that, we have crossing over of genetic material. So some of the genes and some of the genetic material from the paternal chromosome will cross over and exchange itself for a place on the maternal chromosome. So we'll see how they exchange material. And then some of the maternal chromosome will exchange and cross over to the paternal chromosomes. So we get this process of crossing over so that we end up with 4 unique chromosomes that they code for the same thing, but what they're expressing will be completely different. So this is, at the end, each one of these chromosomes will end up in a separate sperm. And so what this ensures is that none of the siblings from these genetic material, from each of these chromosomes, none of the sperm will be able to give rise to siblings that are the same. All the siblings will be different because of this crossing over process. Okay, so now at the end of meiosis 1 then, what's going to happen is instead of like in mitosis where we had the father, so we had the paternal chromosomes and the maternal chromosome together and they would separate and go into opposite cells, right? So we're not going to have that. Instead what happens is both of the paternal chromosomes are going to end up in one cell, and then both of the maternal cells are going to end up in the other cell because the tetrad splits. So we get a splitting of the tetrad, right? That splits, and then we just get both of the maternal in one and both of the paternal ones in the other. Now we still see that effect of the crossing over, where we're going to have the 4 chromosomes just looking completely different from each other. So now, at the end of meiosis 1, we end up with secondary spermatocytes. So here we have the secondary spermatocytes. They both have 46 chromosomes, but they're not pairs. So we're going to call these haploid because they only have half the number of pairs that they should. They do have 46 chromosomes, but they don't have 23 pairs, right? So this is a haploid, whereas at the beginning when we had the primary spermatocyte that had 23 pairs of chromosomes, that was considered diploid, right? So now we have a haploid cell. So let's take a look at what happens to the secondary spermatocyte. So down here, I'm just showing meiosis 2. I'm showing what's going to happen in meiosis 2. We have the chromosomes. There's the paternal chromosomes, and there's the maternal chromosomes. So these are still the secondary spermatocytes. They still have a little bit of their crossing over. They'll always have that genetic material crossed over. So it's always going to be like that. And then what happens during anaphase then is that one of the chromosomes from that secondary spermatocyte will end up in one daughter cell, and the other chromosome will end up in the other daughter cell. So we see that with both. And so we end up with one cell. There's a paternal chromosome there, and then we see the maternal chromosomes there, and they still both have that crossing over. So this is what they look like. So we can see that none of the spermatids here look alike. So this means that each one of these spermatids will become a sperm, and none of the sperm are going to look identical. They're not going to be able to express the chromosomes in an identical way. So again, what are some important things you need to know about this process? First of all, you need to know that at the beginning of spermatogenesis, we start out with the spermatogonium, and after mitosis, we end up with a primary spermatocyte. The primary spermatocyte is diploid, and it has 23 pairs of chromosomes. That primary spermatocyte then goes through interphase 1, where the DNA replicate, and then meiosis 1, and at the end of meiosis 1, we end up with a secondary spermatocyte. The secondary spermatocyte doesn't have 23 pairs of chromosomes, even though each cell has 46 chromosomes. So we say that these cells are also haploid because they don't have the 23 pairs in each of the cells. So the other thing that happens during meiosis 1, and we need to make note that this happens during prophase 1, we get the tetrad formation and the crossing over. Right? And then we end up with a secondary spermatocyte. The secondary spermatocytes then, and there's 2 of those, they are going to each go through meiosis 2. At the start of meiosis 2, there is no interphase. So these cells have the number of chromosomes that they are going to have to split. So then at the end of meiosis 2, we end up with 4 spermatids. Each of the spermatids only contain 23 chromosomes that are all unique from the same chromosomes that are coding for the same things in the other spermatids. So each spermatid has unique genetic material. All right, then let's take a look at what happens to those spermatids. This is just a repeat sort of what I'd already talked about. So the spermatids then are going to, there's 4 of them. Each one of those are going to go through the process that we call spermiogenesis. Spermiogenesis then will give us 4 spermatozoa. So these spermatozoa now look like sperm, but they're immature. They're also incapacitated. Incapacitated means that they are immotile. They can't move. These 4 spermatozoa then are going to go through the process that is called spermiation. Spermiation releases them from the nurse cell. So now they are still 4 spermatozoa, but now they are free in the lumen of the seminiferous tubule. Now these 4 spermatozoa are going to end up going into the rete testis, and from the rete testis they are going to be moved into the epididymis. In the epididymis, they are going to mature, but they're still going to be incapacitated. And then from the epididymis, they'll move into the Ductus Deferens. The Ductus Deferens has cilia, and the cilia will help to move the epididymis through the Ductus Deferens, and we're going to talk more about that in our next video. This whole process, if we look at starting as a 1 spermatogonium, all the way down to the spermatozoa, this whole process takes 9 weeks. And it begins when the anterior lobe of the pituitary secretes FSH. So I'll be talking about the hormones in just a bit. Let's take a look at the anatomy of a spermatozoon. So that is a sperm, a mature sperm. So the spermatozoon has a head, and the head is really just the nucleus, and there's very, very little cytoplasm around it. Then there's a middle part, and then there is a tail. So this is the head, this is the middle, and this is the tail. So the head contains the chromosomes. The middle contains mitochondria, and the tail is the flagella. So this is the only cell in a human being that has a flagella. There are other cells in the environment, like bacteria, that have flagella, but in the human being, only the sperm have these flagella. So the middle part, again, it contains the mitochondria. So the middle part is also, we could say, is the neck. And these mitochondria are going to produce ATP. And it's the ATP that's necessary to make the flagella move. For the mitochondria to produce ATP, the cell has to absorb nutrients from the surrounding fluid. The nutrient that will be the most readily available is called fructose. So fructose is a simple sugar, and that'll get absorbed into the spermatozoon, and then that fructose will be used to produce the ATP.